Good afternoon, everyone. I'm Yuishin KOSAKA from Osaka University, Wataru Aoki Laboratory. Today, I'd like to introduce our research titled Ribosome Self-Replication Outside Living Cell. First of all, I'd like to thank the organizers to invite me here. It's a great honor for me to be here. So I hope I can offer everyone some useful insights to this symposium. Okay, to begin, I'd like to address the core theme of this symposium. What is life and what is non-life, and how to transition. And what truly defines this life and non-life and the transition? One of the most fundamental requirements for life is self-replication, the ability of self-replication. And in our study, we prepared non-life self-re platform by killing E. coli living cell. And in this self-re platform, we reconstituted the very fundamental aspects of life, that is self-replication of ribosome. And in the field of synthetic biology, one of the ultimate goals is the creation of self-replicating artificial cells, or understanding the self-replication of life. In the previous study, researchers successfully reconstituted each step of central dogma. The reconstitution of central dogma is essential for creation of artificial life. And for example, DNA replication or transcription or translation process has been successfully reconstituted. However, for ribosome self-replications, this is a very challenging goal, and it has not been reconstituted so far. And there is a reason how it is challenged to reconstitute ribosome self-replication or ribosome biogenesis. The reason is very simple. Ribosome biogenesis is a very complex process. Even in a simple E. coli ribosome, the related factors is nearly 200. For example, 3 ribosomal RNA and 54 ribosomal proteins are required as a structural component, and nearly 100 accessory factors, that's about folding or maturation and processing of these components, are also required. And nearly 50 transcription or translation-related factors, such as transfer RNA or AARS or translation factors, are also required. And all of these components are coordinately interacted to assemble functional ribosome. So this is how the complexity is and the reason for the challenge of reconstituting the ribosome self-replication. So we prepared 2 strategies. The first strategy is imitating the living cell. We constructed a cytoplasm mimicking reaction solution. Although this is an in vitro system, we imitated the living cell. We used E. coli extract, and we optimized the composition of salt conditions in this experiment system. And the second strategy is the management of resources in this experimental system. Because this platform is not a living cell and closed system, the resource for transcription and translation is limited. So we have to use these resources in an optimized way. So we titrated each DNA template for ribosome components to perform ribosome self-replication in vitro. Okay, let's move on to the first section. First, we developed specific and sensitive detection assay for newly synthesized ribosome. To prove self-replication of ribosome in such an in vitro system, we need such a system, because in this system, we add the starting native ribosome, and using this native ribosome, we express ribosomal protein gene in this cell-free system. And the expressed ribosomal components form newly synthesized nascent ribosome. So in a reaction solution, there are two types of ribosome coexist. The one is starting native ribosome, and the other is the newly synthesized or nascent ribosome. And what we want to detect is only this nascent ribosome. So we have to specifically and sensitively detect this nascent ribosome. To support specific detection, we used orthogonal translation system. In nature, ribosome can recognize messenger RNA using the pairing of specific sequence called Shine-Dalgarno SD sequence on messenger RNA and anti-Shine-Dalgarno sequence, ASD sequence on ribosome small subunit. And previous study, researchers inserted mutations to this SD and ASD pair to support orthogonal translation. If we use artificial orthogonal SD sequence as a reporter, we can cut off the background from native ribosome in a reaction solution. And if we use orthogonal ribosome with orthogonal anti-SD sequence, we can cut off the mistranslation of endogenous messenger RNA. And when we combine these two orthogonal system, we can achieve two-sided orthogonality that is almost completely support the separate translation of native ribosome and artificial ribosome. And from literature, we first selected 7 pairs of SD sequence and anti-SD sequence, and to identify the two-sided orthogonality in vitro system. First, we screened SD sequence. We prepared wild-type SD with fluorescent reporter or orthogonal SD sequence with fluorescent reporter gene. And we added to this gene to reaction solution with native ribosome. The result was clear. When we use superfolder GFP gene as a fluorescent reporter or more sensitive LAC-C reporter gene, we could see that selected the OSD sequence can cut off the fluorescent signal from native ribosome. Moreover, when we tested orthogonal anti-SD sequence, we could see that this orthogonal ribosome cannot recognize this wild-type SD sequence. And this orthogonal ribosome can translate corresponding SD sequence, very high efficiency. And we selected from these pairs OR1 as a best candidate. Then we constructed a sensitive assay to so that we can detect even a very trace amount of nascent ribosome. So we confined the microliter scale bulk reaction solution to femtoliter scale very tiny droplet. And this was the result. We could detect picomolar order ribosome, very high sensitive detection. So in this chapter, we created a specific and sensitive detection assay. With these tools in hand, we challenged ribosome self-replication outside the living cell. As I mentioned in previous slide, we imitated living system. So we used E. coli extract system. We cultured E. coli and disrupted them and performed ultracentrifugation to exclude endogenous ribosome. And we prepared S150 extract. To this extract, we added native ribosome from outside and some plasmid for coding fluorescent reporter gene or ribosomal components. And add some reagent that support cell-free protein synthesis, such as NTPs or amino acid transfer RNA mixture. And we first challenged the reconstitution of small subunit replication. So ribosome comprises two subunits, small subunit for decoding and large subunit for peptide bond formation. And small subunit is relatively simple compared to large subunit. So we selected this as a starting first goal. The reconstitution assays are divided in two reactions. In the first reaction, we expressed ribosomal RNA and ribosomal protein gene simultaneously in the same reaction solution to assemble artificial small subunit that contains orthogonal anti-SD sequence and some antibiotics resistance mutation. At the second reaction, we added fluorescent reportaging with orthogonal SD sequence. And we also added spectrum-mycin antibiotics to suppress native ribosomes small subunit to detect the translational activity of assembled artificial small subunits. And this was the result. We titrated the concentration of ribosomal RNA gene and ribosomal protein gene and native ribosome systematically. And although the initial condition gives off no signal, in some condition, we could detect the translational activity from small subunit self-replication. And we continued follow-up optimization. And the signal was improved. And this signal was so strong that signal from droplet assay almost saturated. In this optimal condition, we also performed bulk microliter scale assay instead of femtoliter droplet assay. Surprisingly, we could detect the signal from small subunit replication only when we add native ribosome and ribosomal protein gene and ribosomal RNA gene simultaneously. The fluorescent signal can be detected. While when we omitted only one of the component, the signal disappeared. From this result, we conclude that we could reconstitute the replication of small subunit. And next, we challenged with the large subunit self-replication, although this is a more challenging goal. The same with small subunit, the reaction was divided in two reactions, ribosome assembly and detection of nascent ribosome. We added, again, ribosomal protein, RNA gene and ribosomal protein gene to assemble artificial large subunit that holds the antibiotic resistance. And the second reaction, we added an antibiotics called clindamycin that kills native large subunit with fluorescent reportaging. And this is the result. Again, we titrated the ribosomal RNA gene and ribosomal protein gene and identified the optimal condition. In this optimal condition, only when we added these components all together, we could detect the signal from large subunit self-replication. While we omitted only one of them in this negative control, the translation activity was disappeared. So in this result, we concluded that LSU self-replication was reconstituted. So our final goal is the reconstitution of full ribosome. So in this experiment, in the first reaction, we added ribosomal RNA gene and full ribosomal protein gene, 21 small subunit and 33 large subunit are ribosomal protein genes. In such a reaction solution, we could assume that there are four types of ribosome: fully native ribosome, blue and blue, and half synthetic, half artificial ribosome that comprises artificial large subunit or artificial small subunit. And this is what we need. This is a full synthetic ribosome that comprises artificial large subunit and artificial small subunit. And we prepared this solution and performed three detection assay. When we performed small subunit detection and large subunit detection, we could detect the translation activity from small subunit self-replication and large subunit self-replication in the same reaction solution. However, when we combined two antibiotics, we could not detect the full ribosome activity. So we selectively purified these nascent large subunit and nascent small subunit from the reaction solution and combined them to this E. coli extract platform. And we added double antibiotics and fluorescent reportage, whether we could detect the translation activity of these free synthetic ribosome. And this was the result. This is only when we combined nascent newly synthesized small subunit and large subunit, we could detect the fluorescent signal. That means translational activity from the droplet. So that means in current condition, we could perform small subunit self-replication and large subunit replication in the same reaction solution. And each subunit can, although we could not detect, but if they are combined, they could form functional ribosome. Finally, I would like to conclude by discussing the path towards self-replicating artificial cells. As I demonstrated, we reconstituted the self-replication of ribosome in vitro, but the efficiency is very low. We could synthesize only 4 nanomolar of artificial ribosome from 80 nanomolar of starting ribosome. So we have to improve at least 6% of improvement. So this is very challenging, but we will try to achieve self-replicating artificial cells. So this is all for my presentation. Thank you for listening. I'd like to welcome your any comment or questions. Thank you very much.
Yes. I can start from asking a naive question. So why is 26% improvement important here?
Okay, I missed some critical logic because in a living cell, to sustain self-replication, we could assume that one ribosome have to replicate one or more ribosome. So in this case, if the starting ribosome is 80 nanomolar, this ribosome should replicate 80 nanomolar of synthetic or nascent ribosome. But in our current condition, only 4 is the maximum. So the gap is to 6%.
Oh, okay. Did you mean 200% or maybe I'm just not doing the calculation right?
Sorry, the logic of
26. But
sorry, I also get lost.
Maybe we can discuss.
Yeah. We can talk later.
Okay.
Well, thank you for a nice talk. I think I just missed, but so your system, I mean the ribosome genes means are all components of ribosome, right?
All components, yes.
I mean,
yes.
So you don't contain some assembly protein or kind of RNA helicase or some protein for the modification of protein chaperone do not contain in your system, right?
Yes.
So I'm really surprised without those components, you can create the ribosome. So what do you think in the real nature world that these are assembly component are essential, or what is the main role of them?
Okay, thank you for pointing out the critical point. Although we do not add such an assembly factor from outside, we use E. coli extract. And we could assume that this E. coli extract contains some of these accessory factors. Although not all of them are functional, but some of them are functional. So we now assume that this endogenous assembly factor in extract is supporting our system. So this is the room for optimization for further study.
Thank you.
Thank you for a nice talk.
Thank you very much.
So you need to improve the efficiency, right?
Yes.
So for the improvement, you have to use more productive self-reach system, or you have to increase the efficiency of the assembling, which
yeah, sorry, I couldn't tell the difference of improvement on self-reach system and efficiency.
did you synthesize enough amount of ribosome protein in this self-reach system?
Yeah, I believe we synthesize ribosomal protein enough to prove this in vitro ribosome self-replication concept, but efficiency is not enough. So in our
So my question
yeah.
is, did you check the concentration of synthesized ribosome protein?
Yeah, we yes, checked the ribosomal protein expression using mass spectrometry.
Okay.
Yeah.
So if the newly synthesized protein, everything goes to the new ribosome, the new ribosome concentration become 18 nanomolar.
Yeah, but in our current data, although the ribosomal protein is enough, but this nascent ribosome comprises of mixture of endogenous and nascent ribosomal protein. So in this aspect, we have to increase the amount, although the amount is seems enough, but we could add more ribosomal protein to.
We can discuss. Right.
Thank you very much. Thank you very much.
And okay, let's take a 10-minute break.